I'm Luke Thompson with NOAA's Atlantic Oceanographic and Meteorological Laboratory and the Northern Gulf Institute in Miami, Florida. Today I'm going to tell you about how to fill in a FAIRe sheet once you've generated it. And we're going to start here with the README sheet, which is the first one. And at the top of the README sheet, it'll tell you about the FAIRe checklist version that was used to generate it, the date and time it was generated, and the names of the sheets in the FAIRe sheet. So project metadata, sample metadata, experiment run metadata, analysis metadata, and there's a taxa final table here too. Now, each of these sheets will have a timestamp and an email next to it, and those will get filled in once you start editing the sheet. It'll show you the last time that the sheet was edited and who edited it, so you can track progress and collaboration across the sheet. There's also a section for the parameters that were provided when the sheet was generated, as well as the requirement levels for each of the fields and some instructions as well. First, we're going to talk about the project metadata sheet. Now, the way data is organized in FAIRe is we have a project, and within that a sample or a set of samples, and within those samples we have experiment runs or libraries. These are the sequencing runs that are done on those samples. And then within that, we have analyses, which are the analyses of the library data, the DNA sequence data. So at the project level, in this sheet, the columns are organized with the requirement code, and then the section to which they pertain, the name of the term, and then the value. And you can provide either data at the project level, which means it's the same value for the whole project, all samples and all assays in your project, or at the assay level, which is if you have multiple assays, a lot of these values may be different between assay number 1 and assay number 2. So you provide the name of that assay in the column, and then below that, the information about that pertains to that particular assay. So things like the information about the investigators and the lab where it was done, those are all going to be the same for the project. But once you get down to information about the assay, you'll see that there's some assay-specific information here. Now, one thing to point out about these assays is that if you use the bebop format, you can pull that information from the bebop protocol in that YAML front matter. And you can also put a link to the bebop protocol in this nucleic acid AMP cell. So that would go here and here. In this case, the author just put a link to the paper, which had the protocol information, but you can also put the Zenodo DOIs for your bebop protocols in this field, nucleic acid AMP, in the appropriate assay column. One of the things about this FAIRe sheet is we use notes, and notes are these little pop-up notes that appear when there's a little black triangle in the cell. You know there's a note there. You can read it, read about the term, its requirement level, its description, the example, and what type of text goes there. These are very handy. These are distinct from comments. You can also use comments, which work like Google Doc comments to make a comment about something you need to work on, maybe tag a collaborator so they can see it, and then when you're done with that comment, you can resolve it. And another thing to say about the spreadsheet is there are these drop-down menus. These will give you options. If there are options available, similar to the Excel template, you can fill in the values as appropriate among the choices. And there's some validation that goes along with those two to make sure that the term you've entered is a valid term for that field. Next is sample metadata. So in this table, this is a more standard style table where you have sample names on the left column, and then all of the values corresponding to that sample proceed out to the right. And there's lots of different information. This is the biggest of the tables in terms of the amount of information that's being asked. If you don't have information for a column, you can leave it blank or delete it if it's not a mandatory column or highly recommended. And again, there's information about what goes in each column in these notes. And one thing to point out is when you see these drop-down menus, you will get some information about the possible values. But if it's a null value, meaning it's either missing, not applicable, not collected, not provided, or restricted access, you can put those values. These are specific controlled vocabulary terms used by NCBI biosample, and so we've put those in here so that you can choose among these choices, and it gives the user some information about why the data wasn't provided. And then we have experiment run metadata. So this is information about the sequencing libraries or experiment runs that you generated from each sample. And in this table, a sample name or SAMP name can be listed more than once if there is more than one library associated with it. For example, if you sequence a sample with the same assay more than once, or you sequence a sample with 2 different assays, in both of those cases, you can list the SAMP name twice, and then the assay name as appropriate, and the libID that you used. If you are sequencing the same sample with the same assay, you need to have a different libID. And in fact, the libIDs need to be unique in any case. So it's a good idea to include the name, some indicator of the name of the assay in the libID. A shorthand will suffice. Then there's information about the library as we go to the right here, and including the file name. This is very handy if you're generating a manifest file, say for QIIME. You have the file name right here, as well as the sequencing run name. And also there are associated sequences. So this is a field where you can list the SRA, biosample, and bioproject IDs that correspond to that sample. Now that we've generated and filled in our FAIReSheet, we can do some cool things with it. We can convert it to different formats, for example, to a QIIME-compatible metadata format, or to an NCBI biosample and SRA format. So the first thing we want to do is download that Google Sheet as an Excel file, and I've done that already, so that is in this folder called metadata. And so the first thing we're going to do is check out this tool, FAIRe2QIIME. So I'm going to clone that from GitHub. It's on the AOML Omics GitHub organization here. And after I clone it, I'm going to go into that folder. And the command is pretty simple. So there's additional parameters if you want, but the main parameters here are the path to the Excel file, the sample name column. We're choosing libID. This allows us to have multiple libIDs for the same SAMP name. So every libID, which will be the sample name in QIIME in the metadata file, will be unique. We then set the path to the sequences. So here it's /data/sequences, but you should change this to wherever your sequence data is located. And then the output directory, which is, we're going to use that same metadata folder where the Excel file is located. So we run that. And now we can go to the metadata folder, and we can see the 4 files that were generated there. I like to use this program called TabView to look at tabular data. So we can look at there's a metadata file for each, both the REAS and the MyFish data. So the V5 and the V5, V6 data, because we want to have all of the experiment run metadata also in that same file with the sample and the project metadata. So that's what's done here. All of the data from those 3 sheets is merged into one, using the library ID as the sample name, and then the rest of the information is following here. So all those columns are combined into one. So you can analyze the data however you want, and all the data is preserved in this single file. We also have a manifest file. This is used by QIIME to figure out which sample names go with which sequencing files. So that's shown here, and it's using that path, that parent directory, and the path that is relevant for this data. So really nice tool here. And then we're going to go back, and we're going to clone the second repository, which is called FAIRe2NCBI. And we're going to go into that folder. And there are two modes to this script. So there's biosample and SRA. So first we're going to do biosample. And when we run biosample, it's going to ask us if we want to use a config file from a previous run. Since this is the first time we've run it, we'll say no. But if you've run this before, you can use a config file that was generated in the first run for your later runs, and you don't have to answer all these questions. But it's going to ask us some questions since we haven't done this before. It also said there's no bioproject accession provided. Do you want to enter the values manually? We're going to say no because we haven't submitted this to NCBI yet. We don't know what the bioproject is. But ideally, you would get a bioproject assigned first, enter that into your FAIRe or your config file here. Then you would generate the biosample data, and then finally you would and then upload the biosample, and then you would get the attributes file, and that's going to have all of the individual accession numbers for your biosamples, and then you can put those into your input file for the SRA. So then the SRAs get linked to the biosamples, which get linked to the bioproject. So it's an iterative process, but we're just going to do it all here together and not provide those numbers at this time. And it's noticing that some of the mandatory columns aren't provided. So we probably could have just provided this in the FAIRe sheet in the first place, but since we didn't, I know that these are seawater samples, so I'm going to say these are seawater metagenome. If they're regular samples, they're seawater metagenome. If they're controls, we'll want to change this to metagenome for those samples. It's asking then for all of the numeric columns, it's going to suggest units for us, and it's going to use the units that are used or recommended by MixS. If we want something different, we can do that, but I'm going to go ahead and say yes, use the preferred unit meter, or I can just hit enter since that's the default unit. That's for this column, maximum depth in meters. Chlorophyll, milligrams per cubic meter, that's great. Salinity, practical salinity units, also great. Temperature, SAMP store temp, degrees Celsius. Size frac, micrometer, temp, degrees Celsius. Do we want to add values in the sample title column? We'll say yes. And it's going to use the geoloc name, the organism, and the sample name to come up with a title for each sample. I'm going to say yes. And now, do we want to add all of these columns to the biosample metadata? We're going to say no because there's a few of these columns that have the word unit in them. And so I want to go through and remove those. So anytime it says unit, because we don't need these unit columns, so 8, 25, 33, and 36. All right, that's done. That was created in the metadata folder. And we can look at that in a minute, but let's go ahead and run the SRA version 2, the SRA mode. Same thing, it's very simple. You just have the FAIRe metadata file and then the SRA metadata file. We don't have a config from a previous run, so we're going to go ahead and just answer all these questions manually again. So do we want to use all libids or only specific ones? We're going to do all. It's an amplicon survey. This is the default, so we'll just hit enter. It's metagenomic, that's the default, so we'll hit enter. It's PCR, so we're going to hit enter. It's the default. And we know this is a MySeq, so we can enter this number 14. And that's been written as well. So if we go to metadata, we now have also the SRA, Excel file and the biosample TSVs. And so if we open up these two files, this biosample.tsv and the sra.xlsx files in Excel, we can see that everything has been filled in the templates. We have all of our sample names here in the first column and all the other biosample information in the other columns. And in the SRA, we have all of our library IDs and titles and so forth. Going forward, we would just need to fill in these biosample accession numbers. And again, if you get your attributes file from after you submit your biosample file, you'll be able to have those automatically inserted when you run this script.